Good morning, student. We are discussing about the nucleus, ultrastructure of nuclear membrane and pore complex. The presence of well-developed nucleus with a nuclear envelope is the principal feature that distinguishes prokaryotes from eukaryotes. The primary function of a nucleus are to store the cell's DNA, maintain its integrity, and facilitate its replication and transcription. The nucleus is bounded by the nuclear envelope or a membrane of karyotheca. In the nucleus, there are four structures present: nuclear envelope or membrane, nuclear sap or nuclear matrix, chromatin, and nucleoli. Nuclear envelope. The nucleus is separated from the cytoplasm by a double membrane called a nuclear envelope. The two membranes are separated from each other by a perinuclear space or a perinuclear space of varying width. Each layer is about 100 to 300 angstrom apart, leaving a discontinuous perinuclear space that communicate with the channels of a cytoplasmic membrane. The envelope is absent for a very brief period only at the time of cell division. Then we are discussing about the structure of envelope. The two membranes of a nuclear envelope are roughly parallel and each membrane is 70 to 80 angstrom thick. Outer membrane has connected with the endoplasmic reticulum and has ribosomes on their outer sides. The inner membrane possesses a crystalline layer, often coated with the filaments and a fibrous structure, some of which extend deeper in a nuclear and the other may attach to the chromatin material. The nuclear membrane are separated by the perinuclear space. The perinuclear space is filled with a fluid communicating with the endoplasmic reticulum. Second point is a nuclear pore complex. It is a main things which we are discussing in a nucleus. The nuclear envelope is interpreted at the intervals by the nuclear pore, which act as a passageway for transport of various type of materials. The nuclear pore complex is an extremely large structure with a diameter of about 120 nanometer. In vertebrates, the nuclear pore complex is composed of 50 to 100 different proteins. The proteins play a fundamental role in the physiology of all eukaryotic cells as they control the traffic of molecules between the nucleus and cytoplasm. The number of pores ranges from 100 to 5 into 10 raised to 7 in a different cell. It seems that the pore nucleus is related to the pore density and the number of RNA molecules being transported from the nucleus. The pore are arranged in a rows or hexagonal cluster. Pores contain some electron dense material and are enclosed by the some annular material which is a granulars. These circular structure are called as annuli. Pore diameter are varies from cell to cell, generally from 50 to 100 nanometer. The matrix of annuli may be amorphous, fibrous, which is digested by the trypsin. The pore complex is a rigid structure below the inner membrane of the nuclear envelope. There is an internal dense lamina called as honeycomb layer. Nuclear matrix consists of nuclear gel and the chromatin. Nuclear gel, it is a highly granular containing fibrous material and it is rich in a protein. It also contains small amount of DNA and RNA and phospholipids. In this region, events like a replication of DNA, transcription and transport of substance takes place. The chromatin. The chromatin is present in the condensed form called as a heterochromatin. Matrix also has a network of a non-chromatin material forming a interchromatic matrix. Then the function of chromatin. Function of chromatin is mainly function is DNA replication, DNA transcription, post-transcriptional process. Then next part is a nucleolus. The nucleus was first recognized by the Fontan in 1874. It is a generally spherical shaped body situated in the nucleus, either in a central or a peripheral region, hence called as an eccentric nucleus. The number of nucleoli in each nucleus depends on the species of the sets of chromosomes present in the nucleus. The number of nucleoli in each nucleus may be 1, 2 or 4. Nucleus is a characterized feature of the eukaryotic cell while it is absent in a prokaryotic cell. The size of a nucleus depends on the synthetic activity of the cell. Thus, the cell of a little or no synthetic activity. Example: sperm cell, muscle cell, and it contain a smaller or no nucleoli. While the oocyte, nervous cells, and secretory cell, which have a synthetic protein or other substances, contain a large size nucleus. Thank you.